In this activity, you will examine the dermal tissue system, which forms the protective outer covering of plant organs. You will observe two types of surface tissues, the epidermis of a leaf and the periderm of woody tissue. These tissues play critical roles in protection, water regulation, and gas exchange. In addition to support, transport, and storage, plants must protect themselves from drying out, mechanical injury, and pathogens. This protection is provided by surface tissues that form the outer boundary of plant organs. These tissues also regulate the movement of gases and water vapor between the plant and its environment. The epidermis is the outermost layer of cells on leaves, young stems, roots, and flowers. It is typically a single layer thick and is coated with a waxy cuticle that reduces water loss. Although thin, it forms an effective protective barrier while still allowing exchange with the environment. Most epidermal cells are flat and tightly packed, forming a continuous protective surface. Guard cells are specialized cells that contain chloroplasts and surround stomatal pores. Trichomes are hair-like outgrowths that help reduce water loss, reflect sunlight, and deter herbivores. Trichomes come in various shapes, and some produce substances. A familiar example of trichomes that produce a substance is cannabis. These capitate-stalked glandular trichomes on the flower buds produce the cannabinoids, like THC and CBD and terpenes. This is typically what is harvested by growers for its medicinal effect. As you watch the animation, notice the tiny pore opening and closing on the leaf surface. This opening is a stoma, and it allows carbon dioxide to enter for photosynthesis while oxygen and water vapor exit. The two curved cells surrounding the pore are guard cells. They contain chloroplasts and can actively change shape. When they gain water, they swell and open the stoma. When they lose water, the pore closes. The main driver for this is the influx of potassium and chloride ions, which creates an osmotic gradient. This movement helps the plant balance gas exchange with preventing water loss. In woody plants, the epidermis is replaced by a secondary protective tissue called the periderm. The periderm forms the outer bark and provides strong protection against physical damage and water loss. The periderm consists of three layers. Cork cells are dead at maturity and have thick suberized walls that form a waterproof barrier. Cork cambium is a meristematic layer that produces new cork cells outward and philoderm inward. Philoderm is a thin layer of living cells beneath the cork. Before beginning your observations, answer this question. Why is the epidermis essential for protecting plants, and how might its structure influence water regulation and gas exchange? Consider how a single layer of cells can protect an entire plant organ while still allowing gas exchange. Think about how stomata, cuticle, and cell arrangement affect water movement and diffusion. Pause and record your prediction. The first sample that we will look at is cork cells, where we will look at the periderm. The cork you are observing in this lab comes from the bark of cork oak trees, the same trees that produce the cork used in wine bottles. Cork oak bark can be harvested without killing the tree because the cork cambium continuously produces new cork cells, allowing the bark to regenerate over time. In woody plants, cork is part of the periderm, the tissue that replaces the epidermis as stems and trunks increase in diameter during secondary growth. These cork cells are dead at maturity and have thick walls impregnated with suberin, a waxy substance that makes them waterproof and resistant to decay. This structure makes cork an excellent natural protective barrier for the tree, preventing water loss, insulating against temperature changes, and blocking pathogens. It also explains why cork is lightweight, compressible, and impermeable, properties that make it ideal for sealing wine bottles. As you examine the cork cells under the microscope, think about how their thick walls, empty interiors, and tight packing reflect their protective role in both the living tree and the cork products we use. Here are some images of cork cells. These are not my images, since we couldn't seem to cut it thin enough to see the individual cells. Notice the regular brick-like arrangement of cells and their uniform shape. Observe the thick, rigid walls of the cork cells. Because these cells are dead at maturity, their interiors appear empty. Their thick walls and tight packing form a protective, waterproof layer. Pause and draw several cork cells. Record their shape and wall thickness and describe how this structure relates to protection. To get a view of the epidermis and stomata, we will go back to the purple heart plant. Although instead of looking at the stem like we did for viewing rhaphides, we will peel off the lower epidermis. Stomata are not evenly distributed over all plant tissues. In most terrestrial plants, the majority of stomata are found on the lower surface of the leaf rather than the upper surface. This positioning helps reduce water loss. The lower leaf surface is usually shaded and less exposed to direct sunlight and wind, which lowers the rate of evaporation from open stomata. In some plants, such as floating aquatic plants, stomata may be found primarily on the upper surface, since that is the side exposed to air. However, in most land plants, placing stomata on the underside of the leaf is an important adaptation for conserving water while still allowing gas exchange. You will not find stomata in tissues like cork or other woody surfaces because these tissues are designed for long-term protection, not gas exchange. Here we have the purple heart lower epidermis. Under low power, scan the surface and look for lighter or greenish areas where stomata may be present. At higher magnification, locate the stomatal apparatus. I think they look like lips with cheeks. You should see two curved guard cells surrounding a central pore, the stoma, embedded among ordinary epidermal cells. In these images, most of the stomata are closed since the plant was cut from the mother plant in order to prepare the slides. So since it no longer had access to water, it closed the stomata, although there are a couple of open stomata. Pause and sketch the stomatal apparatus. Label the guard cells, the stomatal pore, and the surrounding epidermal cells. You may notice that the guard cells contain many chloroplasts and wonder why that is. Chloroplasts carry out photosynthesis, which occurs in two main stages, the light-dependent reactions and the Calvin cycle. During the light-dependent reactions, light energy is used to produce ATP, a molecule that stores usable energy for the cell. In most leaf cells, that ATP is used in the Calvin cycle to convert carbon dioxide into sugars. In guard cells, however, much of the energy is used for a different purpose. Guard cells actively regulate the opening and closing of the stomatal pore by pumping ions in and out of the cell. These ion movements drive the uptake and release of water, exchanging turgor pressure and causing the guard cells to change shape. Because this process requires large amounts of energy to power proton pumps and other transport proteins, guard cells contain many chloroplasts that supply the ATP needed to control stomatal movement. This activity shows how plants balance protection with interaction with the environment. The epidermis allows gas exchange and limits water loss, while the periderm provides strong long-term protection for woody tissues. Once again, structure reflects function, from thin, flexible epidermal layers to thick, suberized cork cells.